Nice weekend, huh? How was the exam? Better? Okay. People adjusted to this new scheme a little better? Comments about it? No comments about it? Is that what the final is going to look like? Yes. Except for there'll be different questions. Yeah. Well, I might surprise you and make them the same questions, wouldn't that? See how much you learn? What's that? I'll tell you what I do in the final sometime, and I'll do it this time, is I can let you write part of the final. You can submit questions, and I'll pick 2 or 3 student questions for the final. How's that? No? Students are too rough on each other? How many would like to be able to write part of the final? How many would not like to be able to write part of the final? Oh, okay, then we won't do it. That's really unusual. Most classes decide they want to do part of that. That's interesting. You guys just don't trust your fellow students, do you? Okay, then we won't do that. The final will be like the last final. It will, that is not in the format, but in terms of having a note card, you will get a note card for that. And you will need to get the note card from me. And if you don't have a note card, you will lose points. So it's important to note all of that. I'll make the note cards available later. We're in the home stretch right now where there's nothing between us and the end of the term except for a few lectures and the final. So that's looking up. Okay, well, I want to finish up a couple things today about DNA repair and recombination, then move on to our next topic, which is transcription. And this won't, the finishing this part up won't take but just a few minutes. I talked last time about several different types of DNA repair. And DNA repair is very important for cells to be able to undergo. Understanding that damage is going to happen no matter what we do, and errors are going to happen no matter what we do. Having repair systems to help to alleviate that damage or alleviate those numbers of mismatch errors that occur helps cells to maintain the integrity of their genome, and that's very important. With respect to DNA repair, there are at least a couple of other things I want to mention. One is, I'm not going to talk about Huntington's disease. If you're curious about that, come and see me and I can tell you more about that. That's not really a damage as such. It's actually an unusual type of error that happens in replication. But I do want to mention that some of the different things that can happen relative to error. We've been talking a lot of times about how different errors and so forth can lead to mutation, and how mutation can lead to cancer. A really good example of something that can lead to cancer is this disease called HNPCC. It's actually the most common type of inherited colon cancer. And it is a major contributor to the overall incidence of colon cancer in the United States. HNPCC can be inherited, and it is known that HNPCC, the mutation that causes HNPCC, affects DNA repair. It specifically affects DNA repair. So it's quite clear that if the DNA repair system is not functioning properly, that can lead to mutation, and that mutation can lead to cancer. So it's not something that's theoretical. It's actually fairly well known. The same is true of certain types of breast cancer as well. So mutations that affect the ability of the cell to repair DNA do lead to other problems. I talked last time a little bit about P53, and P53 was what I said was the control, or the quality control for the cell. P53 plays the role that I described to you, and what I didn't get to tell you was that P53 is very commonly mutated in cancers. So if you mutate P53 to where it doesn't function or doesn't function properly, then that quality control is gone. So that when quality control is gone, it's much more likely that errors will slip through, because P53 is reducing the likelihood of cells being able to survive errors. And so if P53 is mutated so that it can't function, many more cells will survive after they've had errors or damage to their DNA. and those damaged DNAs in those cells can lead to uncontrolled growth in cancer. So not surprisingly, p53 damage is very common in many types of cancer. And the last thing I want to say about DNA repair and damage is the Ames test. The Ames test is a very cool type of test. So let me tell you a little bit about how it works. With the Ames test, you would like to be able to detect the rate of error. What is the rate of error or the rate of damage or something that's affecting the ability of the cell to properly replicate its DNA? Okay? You'd like to know what that rate is. Well, I've told you so far that that rate is very, very low. Very, very low. Okay? So for a bacterial cell, for example, the rate is on the order of about 1 in 10 million base pairs. So I have to have an assay, if I want to study it, I have to have an assay that is going to tell me something that is so sensitive I can detect 1 in 10 million. Okay? Well, the way this is done is actually kind of a cool scheme, and the person who designed it, Bruce Ames, really deserves a lot of credit for a very cool assay. The idea goes as follows. Let's imagine that I have a gene, and that gene is able to kill bacterial cells. So that if bacteria have this gene, okay, they will die. All right? If they have this gene, they will die. So I take this gene and I put it into a plasmid, and I transfect it into bacteria. And what would I expect would happen with that? Would I expect to see living bacteria? Wouldn't expect to see any living bacteria. If I see living bacteria, what do you suppose has happened? Mutation that has affected that gene, right? Well, I specifically set it up so that this gene can be mutated in one place, and that one place will cause it, the gene itself, to not be able to function, right? So I ask the question, how often does that mutation occur? How often does that mutation occur to knock out that gene? All right? So I take a batch of cells. I might take a batch of cells that might have, let's say, 500 million cells. It wouldn't take a lot of cells to have 500 million cells. I transfect those cells with this plasmid, and I spread them onto a plate, and I count to see how many of those cells lived. Right? When I count to see how many of those cells lived, would I expect to see some cells? Yes, I would, because some mutation is going to happen. Right? So if I count the number of cells, I get an indication of how much mutation is happening in those cells. Well, that's not why the Ames test is really useful. The Ames test is useful because if I compare one batch of cells that have been untreated with a chemical, for example, and another batch of cells that have been treated with a chemical that I think might cause DNA to mutate, then if I compare the counts of those two cells, I can quickly answer the question, which one has more mutation? Right? So if I look at the cells and I see that the cells that have been treated with this chemical have more living cells than those that didn't, I know that that chemical that I've treated them with is favoring DNA mutation. Okay? Does that make sense? So I have a selection that's quite simple, and all I have to do is compare number of counts of living cells. Right? So if I see that I've got 3 times as many cells living in the treated versus the untreated, I say, whoa, okay, I have to be worried about this because this compound is a mutagen. It favors the mutation of DNA. Okay? This is shown in another way on this plate. On this plate over here on the left, you see cells that were untreated. That is, they had no addition of chemical. But on the right, there's a chemical right there in the middle of that little dot. And you can see what's happening around that dot is that many cells are now living that weren't otherwise living. That chemical that's in there is favoring mutation and favoring the stopping of function of that gene that's killing the cells. Make sense? There's many ways in which the Ames test can be done. I've described one to you. We could do it with color indicators. We could do it with a variety of things. But this particular one that I've just described to you is a very effective way of doing the Ames test and understanding how mutation affects cells. Okay. Well, the last thing I want to briefly mention is recombination. And recombination is a way of rearranging the DNA that's inside of cells. Okay? The rearranging the DNA that's inside of cells. Okay? So schematically, we see it as follows. We have two separate strands of DNA that's up at the top. All right? And these sequences of these two DNAs might be very, very, very similar. If we put these two sets of DNA next to each other inside of cells, and yes, that actually happens in our cells during the process of cellular division, where the homologous chromosomes lined up, what we see is that there can be what's called a crossover event between those two, so that we get a mixing of the DNAs from one DNA to the other. This event that occurs is relatively rare, but it's happened in every one of us, and it's happening in every one of us right now. So when we think about mutation, and we think about changes in DNA, we also have to keep in mind that recombination is another thing that's affecting DNAs. And that recombination is giving rise to new DNAs that didn't exist previously. Okay? That's not unlike what we talked about with respect to biotechnology, where we were taking a plasmid and we were ligating into it a foreign sequence. We call that recombination. This is cellular recombination. So recombination is happening in nature just like it can happen inside of a test tube. Okay? And that recombination gave rise partly to you. Because of recombination that happened very early during or at the time you were fertilized, okay? The egg cell that was fertilized that gave rise to you, okay? Recombination happened at that point and gave rise to DNAs that were in neither of your parents. We always think about the fact that we're the product of our two parents. But in fact, we're the product of our two parents with a little bit of mixing of DNA that happened as a result of recombination. Okay? So recombination does provide new mixes of DNA that didn't exist in either parent. There are a variety of ways in which recombination happens, one of which is shown here, a process involving strand invasion, and it's actually showing you how homologous sequences actually can fit with each other. So we see that this purple strand is actually invading this duplex over here, and because it's homologous, it can form reasonably stable base pairs with the blue strand, as you see here. They may not be perfect base pairs. There may be a few mutations here and there, all right? But if they're very, very similar, most of the base pairing will occur as before. This structure gets resolved, and when the structure gets resolved, it forms something called a Holliday junction. And no, I'm not going to expect you to memorize how this occurs. Okay? Holliday junctions arise during recombination, and they arise with what's called a cruciform structure, meaning it's a cross. So we see the two separate strands here, the one on the top, the one on the bottom. We see the strand invasion occurring between the two, and that strand invasion is leading to a cruciform structure, a cross. There's one part of the cross, there's the other part of the cross. Okay? That gets resolved, and when it gets resolved, we end up with two strands not going top and bottom, but now going left and right. And as a result, we have brand new DNA sequences that have been brought into conjunction with each other. Okay? There are enzymes that favor this process, okay? One of which is called a recombinase. Recombinases help facilitate the process of recombination. And there are viruses, for example, that have enzymes like recombinases. Now, when you hear about a virus that has an enzyme like a recombinase, you might think, hey, maybe this virus is going to favor recombination into cellular genomes. And that's exactly what happens. HIV is a prime example of this. In fact, most retroviruses have an enzyme that looks like a recombinase. HIV has an enzyme called integrase. And integrase helps facilitate the integration of the viral DNA into the genome. That's how the HIV virus DNA gets into the genome. Yes? Okay, so his question is that why cancers are more common in HIV patients than not? To a limited extent, yes, but more commonly the cancers that are occurring in HIV patients are due to the fact that the immune system is compromised. So the immune system compromise leads to more cancers, and that's a very well-known phenomenon. Your immune system is protecting you against cancerous growth in many cases. So if you compromise the immune system, you will see more cancers. But this, to a limited extent, will contribute to that as well. Okay? Okay. Yeah. Yes. Say it again. Okay, so his question is that I said that these lead to mismatches, and when these mismatches occur, which strand is used as the template for repair? Either can be. Either can be. And so we see that the resolution of that can be then random, and that random is what favors the mutation and the changes that are happening with this. So it's not like the situation we had where we could tell which strand was the new and which strand was the old. We can't tell it here. So this is going to be a random resolution that's going to happen. Which one's going to be the template? Okay? Yes? Okay, how can I say it's relatively rare, but it happens all the time? Ahern, you're not being consistent is basically what she's saying. Okay? It is relatively rare, and it happens all the time. Keep in mind, you have several trillion cells. Relatively rare, if it happens once in a billion cells, is still happening thousands of times inside of your body. So it's relatively rare, but it is happening all the time. So I wasn't just bullshitting you. Okay? Yes? Say it again? Will topoisomerases play a role in here, I think, is your question. And the answer is, yes, they do. Topoisomerases do play a role in resolving these structures and in helping these structures to even form. So yes, you're exactly right. Topoisomerases do play a role in genetic recombination. Yes? Is this happening only in meiosis or only mitosis? This can happen anytime DNAs are brought into close proximity with each other. Yes? What's that? What does the cell do with this after it happens? Well, we could imagine, as the question over here was, we have mismatched strands. So what is the cell going to do with that? The cell is going to try to resolve those, and that resolution of those mismatches is going to lead to mutation. Why do cells do this in the first place? Because over evolutionary time, there is advantage to mutation. The advantage to mutation is those mutations which lead to death will be selected against, and those which give rise to new traits that are beneficial will be selected for. So this is actually a part, when we talk about evolution, this is part of an evolutionary process. Just as mutation itself is part of the evolutionary process, so too is recombination. So that's the importance of it in terms of overall life on Earth. Good questions. Okay. That's the last of what I want to say about DNA repair and recombination. I'd like to turn our attention now to the next topic, which is transcription. And talking about transcription, we're turning our attention from a DNA-making process now to an RNA-making process. And that means that instead of talking about DNA polymerase, we talk about RNA polymerase. And RNA polymerase is different. RNA polymerases are different from DNA polymerases in several ways. Okay? Now, I talked earlier about primase being a RNA or an RNA polymerase. All right? It is not the RNA polymerase that is used to make RNA in cells. The primase is used only to make primers. It's not used for any other purpose. So when we talk about transcription, all right, primase is not something that's used to make RNA like messenger RNAs and so forth for cells. RNA polymerase that exists inside of cells, okay, exists with a structure that looks not unlike what we saw for DNA polymerase. Remember DNA polymerase had that hand that looked like this that held on to the double helix? Well, we can imagine there's that hand again that's holding on to that double helix. Okay? And actually, in RNA polymerase, it's actually holding on to one strand. It's not holding on to both. Okay? And that's because when we make RNA inside the cell, only one strand of the DNA is copied. Right? DNA polymerase had a real job. It had to look this way, it had to look that way, it had to do leading and lagging strand synthesis. RNA polymerase doesn't have such a complicated life. RNA polymerase is only copying one strand. Now you might think, therefore, that RNA polymerase can go zipping along a lot faster than DNA polymerase would because it's got less to do. In fact, RNA polymerase goes much less fast than DNA polymerase does. There's a conundrum for you. Okay? It goes much slower than DNA polymerase does. Okay? Where DNA polymerase was moving along at the rate of about 1,000 nucleotides a second, in E. coli, and that's where the 1,000 nucleotides a second is occurring, okay? In E. coli, RNA polymerase is moving along at the rate of about 50 to 60 nucleotides a second. Way slower. Way slower. Now, there are some reasons why that occurs, and I'll talk about those as we get going along. But RNA polymerase is not moving along like DNA polymerase. Another thing I should tell you is that in bacterial cells, there's only one RNA polymerase. One RNA polymerase, if we exclude primase. Primase is a separate category. There's only one RNA polymerase that's making all the different types of RNA. In bacterial cells, we see three types of RNA. We see transfer RNA, we see messenger RNA, and we see ribosomal RNA. Okay? Now, we'll talk about those RNAs as we get going along. I want to say a little bit more about the RNA polymerase. Okay? The RNA polymerase is a multi-subunit protein, and we're not going to talk about functions of the individual subunits except for this guy down here, the sigma subunit. Okay? I'll talk about that function in just a little bit. But before I talk about the subunits and the function, I want to tell you some differences between RNA polymerase and DNA polymerase. Okay? One difference you've already seen in primase. Primase did not need a primer to start making RNA. And RNA polymerase that makes the messenger, the transfer, and the ribosomal RNA does not need a primer either. It works 5' to 3', just like a DNA polymerase does. I've noted that it moves much slower. And RNA polymerase, in conjunction with some other proteins, can do a sort of proofreading. It's not the same as it is in a DNA polymerase, and if I have time, I'll talk about that very briefly later on. Okay? But it's not moving as fast as a DNA polymerase is. Okay. When we look at the synthesis of RNAs off of DNA, as I said, the strand is synthesized 5' to 3', which means it's copying a strand that is oriented 3' to 5' because antiparallel strands are how nucleic acids are made. We have names that we give to the individual strands of DNA. Okay? We have a strand called the coding strand. The coding strand has the same sequence as the RNA that's made, except for U replaces T. The coding strand is not the one that's copied. It's the other strand that's copied, and that's why the coding strand is the same as the RNA transcript, because the coding strand was originally made from the template strand anyway. The template strand is the strand that's being copied by the RNA polymerase. So we can see A pairs with T, A pairs with T, C pairs with G, G pairs with C, U pairs with A, etc. This guy would be made from left to right, from 5' to 3'. That would mean that the red one is 5' to 3' going from right to left. Okay? Another thing that you see on this figure is a sort of nomenclature that we use for RNA is the place where RNA synthesis starts is called +1. That's the very first base. That's where the first base is put in. We call that sequence +1. And if we look in the DNA, the base that's before it is -1. So this would be -1, moving leftwards, -2, -3, -4, -5, etc., etc. So later I'll be talking about the -10 and the -35 sequence. And when I talk about those sequences, I'm talking about that number of nucleotides ahead of, okay, in this case, to the left of the place where synthesis starts. I say it's ahead of because if it's leftwards and synthesis is moving rightwards, then they're opposite to the direction that synthesis is occurring. Okay? All right. So keeping that numbering scheme down is very, very important. Okay. RNA polymerase, like DNA polymerase, uses triphosphates. In the case of RNA polymerase, it's using ribonucleoside triphosphates, not deoxyribonucleoside triphosphates. That means it's using ATP, UTP, CTP, and GTP. Okay? There's no TTP that's involved in RNA synthesis. All right? During the replication process, okay, the same base pairing rules that we saw before are used to make the RNA as we're used to make the DNA. And we also see clipping off of 2 phosphates and incorporation of 1 phosphate into the growing strand. Okay? Now, this shows some mechanism I'm not going to talk about. Here's the 2 phosphates right here being clipped off of the strand. Off of the nucleotide as the rest of the nucleotide is being incorporated into the growing strand. Okay. Yeah, it's more mechanism, and that is more mechanism. This actually shows us something that happens. One of the reasons RNA polymerase is slower than DNA polymerase. RNA polymerase moves in what we call fits and starts. That is, it will go for a ways, and then it'll stop. And it'll actually back up a little bit. It's not unlike what the DNA polymerase is doing when it's proofreading. That backing up allows other proteins to come in and look to see, is everything here properly matched? That's how proofreading actually occurs during transcription. Proofreading during transcription is not nearly as good as it is in DNA polymerases. Transcription is much more error-prone than is DNA replication. But the consequences of errors in making RNA are not the same as they are in making DNA. DNA is passed from one cell to the next to the next. It is the integrity that the cell has, the blueprint of the cell. RNAs are used to do specific things, make proteins. Okay? If you make an error in making one RNA, it's very easy to just degrade it and make another. In fact, cells are doing that all the time, making RNA and breaking it down. Making RNA and breaking it down. So the consequences of errors in transcription are not nearly the same as they are in DNA replication. Another thing about transcription that's different from DNA replication is transcription starts at specific sites we call promoters. In DNA, we call the start place an origin, so the names are different. But we see that promoters are fundamentally different from origins of replication. Okay? Promoters vary. So if I have a gene, and a gene is a segment of DNA that codes for protein, if I have a gene and I want to transcribe, that is make messenger RNAs from that gene, I have to have something in the cell that says, hey, dummy, go over here and start making RNA. That something is a promoter. That promoter has a specific sequence, and that sequence is recognized by the sigma factor of that RNA polymerase I showed you earlier. Now, please note that when I'm talking about transcription here, I'm talking about transcription in prokaryotic cells. Eukaryotic cells, which we'll talk about later, their transcription is way more complex than prokaryotic cells. In prokaryotic cells, it's relatively simple. Okay? So the sigma factor of the RNA polymerase helps the polymerase to recognize the place where to bind to the DNA and start making RNA. So that's why it's also important that a promoter be adjacent to where transcription is going to start. Because obviously you don't want to be randomly making RNAs. You want to be making RNA that wouldn't be efficient for the cell. You want RNAs to be made specifically at the place where they are needed. So having specific sequences that flag that for the cell are important. Now if you look at this, what you can see on the screen, A, B, C, D, E, is you see the -10 region of 5 different genes. So 5 different genes, here's where their start site is. And in the -10 region, we see that there's a region of sequence that's TA rich. Okay? This is TA rich. The -10 sequence is a recognition site for the sigma factor. Now, why do they have different sequences here? Well, they're actually quite similar. If we look at this, TAT, GTT, TAT, GGT, TAA, CTA, okay, they all have a common feature of being AT rich. And if we look over many genes and we average the -10 sequences, we come up with a sequence, like what's called a consensus sequence, that the most common average sequence is TAT-AAT. I still haven't answered the question about why they vary. Okay? Well, it turns out that if we examine these inside of cells, when cells make RNA, they don't make just one copy of an RNA, usually. They make many. And the more copies of a messenger RNA that they make, remember that messenger RNA is the coding for the protein, the more copies of messenger RNA that they make, the more protein can be made. Now, we start thinking about what are the needs of a cell. You've already seen, for example, in DNA polymerases, you saw that DNA polymerase 1, there were about 1,000 copies of DNA polymerase 1 in a cell. And there were only 4 to 6 copies of DNA polymerase 3 in a cell. Cell didn't need 1,000 copies of DNA polymerase 3. Would you imagine, therefore, that you would have more messenger RNAs for DNA polymerase 1 than you would have for DNA polymerase 3? Yes, you would. Okay? So how does the cell control how many copies of a given messenger RNA that it has? Well, one of the ways in which that can occur is by having different promoters in front of genes. One of the ways is that. It turns out that if we compare the number of messenger RNAs made and to the sequence that's in front of those, what we discover is that the closer the sequence is to the consensus sequence, the more messenger RNAs are made. And the more it differs from that consensus sequence, the fewer messenger RNAs are made. Why is that the case? Well, it's the case because it turns out that proteins, like the sigma factor, have to bind to a sequence. Their favorite binding target is right there. They're going to bind this sequence with high efficiency. They're going to bind this sequence with a lower efficiency. Less binding, less transcription, fewer messenger RNAs. Okay? Now, the -10 sequence is only one of many sequences that affect transcription. This one is really important. Others vary somewhat. Okay? Others vary somewhat. Before I move on to those others, any questions about this before I move forward? I'm doing a lot of talking here. Questions? Question? Over here. Oh, yeah. Question? This is specific to E. coli. That's correct. But we could extrapolate this to basically all prokaryotic cells. But eukaryotic transcription will be very different from this. Prokaryotes will all be very similar to this. Yes? Yes. So his question is sort of restating what I said earlier. That is, the more that the sequence differs from the TAT, the fewer messenger RNAs will be made. That's exactly right. Yes, up there. Okay, so his question is, after the sigma factor binds to the sequence, basically what happens, right? Okay, so since that's sort of the next thing I'm going to talk about, I'll address that right here, okay? It's not a coincidence, first of all, that the sequence that we're talking about here is AT-rich. AT-rich sequences, you recall, have fewer hydrogen bonds that hold the strands together. We can imagine then that it's easier to separate these strands than it would be if this sequence was not AT-rich. So one of the things that's going to happen, to answer his question, is that the strands have to be pulled apart, and then the polymerase has to start copying one of them. Okay? The strands have to be pulled apart, and the polymerase has to start copying one of them. In the process of copying one of them, the sigma factor goes bye-bye. It's done its thing. It's bound. It's helped the polymerase to recognize where it needs to be. Now it lets the polymerase go and do its thing. Okay? Other questions about that?
Is this on the coding strand or on the antisense strand?
It is what on the coding strand? So this right here is on the coding strand. You can see it's going 5' to 3'. So what's going to be copied is going to be on the complement to this right here. Yep. Yes?
Do you have multiple copies of a gene in your genome? Does it always have the same promoter before all of them?
Okay, so her question is, if you have multiple copies of a gene in a genome, does it always have the same promoter in front of all of them? In E. coli, you will generally not see the same gene duplicated. You might see homologous things to it, but you won't see the same gene duplicated, because if you have duplication, what you will see is recombination. Because that pairing of strands that I talked about before doesn't have to be on separate DNAs. A DNA could loop back upon itself and align similar sequences. So we don't generally see in prokaryotic cells multiple copies, identical copies of the same of genes. Okay? When we talk about eukaryotic cells, we do see some multiple copies of genes, and again, that's more complicated, but I'm going to save that to talk about later. Okay? Yes? The promoter sequence, as written here, is on the coding strand. That's correct. Well, the template strand would simply be complementary to that, right? So we just do it. We write it in the coding strand. That's just the convention that we write it. But we could just as easily write it on the template strand. We would have to write it 3' to 5', right? Yes?
The Z strand also has a DNA section right after that one.
Yeah.
So is it going to have a different transcription slot or farther down?
Very good question. Okay. So look at this guy right here. That looks like a Tata box, right? I shouldn't say Tata box because that's actually used for eukaryotes. But it looks like a -10 sequence, right? It looks like this sequence here. His question is, will the sigma factor recognize this and initiate transcription 10 base pairs down from this? And to some extent, yes, it will. To some extent, yes, it will. So we see what are called alternate start sequences, and those alternate start sequences can, in some cases, lead to entirely different proteins. Okay? We'll talk about that a little bit with respect to translation, but very good observation. Okay. So there is transcription. There's another sequence that is commonly found near the start sites in E. coli cells, and it's called a -35 sequence. There's a -35 sequence. And -35 sequences vary more than the -10 sequences. Okay? We see more variation, particularly in the location. It's not exactly 35, and this isn't exactly 10. There's a little bit of fluctuation between different -10 sequences. But -10 sequences will usually be within 1 or 2 base pairs of that -10. The -35 might be as many as 4 or 5 base pairs away. Okay? No, you don't have to memorize the minus 35 sequence. I think you should know the consensus sequence, though, of a minus 10. I think that's a pretty cool thing. Okay. Now, I've talked about sigma factors. Let's consider sigma factors in a little bit more depth. All right? Notice I said sigma factors. That means there's more than one sigma factor inside of a cell. Why would a cell need more than one sigma factor? Well, it turns out cells need different proteins under different conditions. All right? A really good example is what we call heat shock. Okay? Heat shock. Heat shock is a phenomenon that occurs to all cells. Your cells, bacterial cells, you name it. When you have a fever, your cells are experiencing heat shock. When you're sitting out there baking on the beach, your cells are experiencing heat shock. At least your skin cells are. Okay? Heat shock is very, very important for us to consider because what does heat do to proteins? Denatures. And some proteins are very, very sensitive to changes in temperature. Okay? E. coli cells, for example, can tolerate temperatures up to about 42 degrees centigrade, roughly 110 Fahrenheit. Okay? You take them beyond that, they can't recover. But there's that range from 37, which corresponds to our body temperature of 98.6, up to about 110 where they can survive. And they can survive because if you raise the temperature up as high as about 110 degrees Fahrenheit, or 42 degrees centigrade, right? What you see is that they recover by making proteins that help back up on this. They recover by making special proteins that help the denatured proteins to renature properly. Okay? So we talked, I believe last term, a little bit about chaperones, right? Everybody remember chaperones? Chaperones were proteins that help proteins to fold properly. They were originally described as heat shock proteins because what people saw was that these proteins were synthesized preferentially after the cell had been experiencing heat shock. And that gave some clue to their function. The clue being maybe they have to do with keeping the protein natured properly, that is, folded properly. So chaperone proteins do that. When do cells need chaperone proteins the greatest? After a heat shock. Now, if I make a lot of chaperone proteins all the time, or an E. coli cell makes a lot of chaperone proteins all the time, and it doesn't need a lot all of the time, it's wasting energy. So E. coli cells make a little bit, just as much as they need, with a relatively inefficient promoter. But when heat shock occurs, they want to crank up and make a lot more heat shock proteins. Well, if we look at the promoter ahead of heat shock genes, we see, boy, that sucker doesn't look very much like a Tata box. Normally, this guy is being transcribed at a very low level. Very low level. There's very little heat shock protein messenger RNA that's made. However, when heat shock occurs, one of the things that the cell does is it starts making a different sigma factor. The different sigma factor preferentially recognizes, in this case, that promoter right there. Well, what's going to happen when I've got a sigma factor that recognizes this and not this? What's going to happen is genes that have this sequence are going to be preferentially transcribed, and I'll make a lot more of them. Exactly what I need, because this promoter is placed in front of genes necessary for recovering from heat shock. Okay? So alternate sigma factors allow cells to adapt to varying environments. The cell is adapting to a varying environment. And that heat shock, the sigma factor that's specific for heat shock is made primarily when heat shock occurs. Okay? Here's another one, when nitrogen starvation occurs. When the cell is needing to conserve its nitrogen and work with what it's got, a sigma factor that's specific for this sequence is made, and a different class of genes is made than would normally be made. Again, only when the cell is needing that. Now, sigma factors, question? What's that? How will this sigma factor be made under heat shock? It's a very good question. Okay, so the question is, what induces that thing to be made? All right? I can't give you a specific answer, but I can give you a general answer. When we look at genes that are induced under specific conditions, like let's say heat shock, there has to be something that's odd about them that would be sensitive to heat. Okay? Let's think about what that might be. Could you guess one? What would be sensitive to heat that might favor transcription of a specific gene? How about a long stretch of A's and T's in the promoter? A long stretch of A's and T's might be much more likely to come apart and favor the entry of a polymerase. Okay? That's one example. I've talked earlier about how topoisomerases can be induced by the superhelical tension in DNA. More superhelical tension, more unwinding, and again, more entry of RNA polymerase. So cells have a variety of schemes like that that are sensitive to the environmental conditions that allow specific genes to be made. That's one example. Okay? Very good question. Yeah? Yeah? Yes? Well, okay, so he's asking about the Ames test and how the Ames test might be affected, I think, by some considerations here. There are things that affect Ames test. The Ames test is not the sole answer for how the mutation rate is occurring. It's an indicator, but it's not an absolute indicator. Okay, let me talk to you about that separately. I'm not quite sure what you're saying, and I want to just finish up here. I'll talk to you about that later, okay?
As far as sigma factors go, are they regulated exclusively by the environment? Okay. What I'm wondering is if you know what sigma factor you need to increase transcription genes, can you just dump that in absence of the environment that would normally create it?
So her question is, could you basically artificially induce a gene to be expressed by putting some factors in there that would cause that to happen? The answer is yes, you can. We're going to have some examples of that that's going to happen later with transcription. You bet. You bet. Other questions? I didn't mean to stifle discussion there. Okay.